Hello all, and welcome to Methods May Vary, a channel of InfoVoice. This is JP Murray here to report to you another fun fact about some random part of science. But first, to discuss some logistics. As some of you may be aware, my podcast segments say at the beginning, a matter of science. That was the name I was going to go with for my channel. However, after much consideration, I have decided to go with Methods May Vary, mainly because I can be quite spontaneous, and also because it sounds just that more bodaciously attractive than the other. With that aside, time to get into the purpose of this podcast. Last time I went into the elements of the human body. After doing that podcast, I cracked open my Human Anatomy book again, and began to dive deeper in a microscopic view of what the human body is composed of. This brought me to cells and the workings thereof. After rediscovering my love for structure, I realized something. There is absolutely no possible way that I will adequately describe everything about the cell to you. There is just so much to learn and so much to know. It is remarkable how much the cell does for the body. In order to attempt to start the process of learning the cell, I will describe various structures and their functions. First structure to start with should be the plasma membrane. The plasma membrane is composed primarily of phospholipids. A phospholipid is part hydrophobic and part hydrophilic. The hydrophobic portion does not want to be in contact with water, whereas the hydrophilic portion does not mind being in contact with water. This helps the lipids connect with each other and not fall apart when engulfed in water. Throughout the membrane, there are various proteins embedded in the surface to control the flow of substances in and out of the cell. Next up is the nucleus. The nucleus is the housing unit of DNA of the cell. When the cell is divided, the DNA is what offers the building data required to split the cell into two and have each cell form each individual organelles independently again. Other than providing the building blueprints, the nucleus also functions in the synthesis of protein required throughout the cell. Now onto the smaller structures of the cell, the cytoplasmic organelles. First, I should define cytoplasm for you. The cytoplasm is the material within a living cell, not including the nucleus. Ribosomes are structures that serve as the site of protein synthesis. They vary in size and are usually free-floating within the cytoplasm, unless they are attached to rough endoplasmic reticulum. The rough endoplasmic reticulum, or RER, is primarily responsible for synthesizing proteins and transporting them. There is also something called the smooth endoplasmic reticulum, also referred to as the SER. This organelle is responsible for manufacturing lipids and carbohydrates, also stores calcium for cells, and detoxifies harmful chemicals. What the RER transports the proteins to is called the Golgi apparatus. The Golgi apparatus modifies the proteins and ships the proteins for internal use or for secretion through the plasma membrane. Next important organelle to know would be the mitochondria. The mitochondria is a spherical thread-like or rod-shaped structure that's surrounded by a double membrane. Inside the mitochondria are projections that are called cristae. The function of the mitochondria would be to synthesize ATP when oxygen is present. For those of you who do not remember what ATP is from my last podcast, it is adenosine triphosphate, the primary energy source of cells. Other parts of cells that are located along the outside of the cell are cilia, flagellum, and microvilli. Cilia are an arm-like structure located along the outside of the wall. The purpose of cilia would be to move materials along the surface or of the cell. Flagellums are used to propel a cell. Much like you and I would use our legs and arms to help us swim, a cell uses flagella to propel itself. The microvilli are simply used to increase the surface area of a cell, making it more possible to absorb liquids. A microvilli may also be modified to form sensory receptors. Now that I have described the wonders of the insides of a cell, I would like to describe the process of division. When a cell divides, there are clear stages that a cell goes through. The stages are as follows: interphase, prophase, metaphase, anaphase, and telophase. To start with is interphase. Interphase is when the cell is functioning like normal, or beginning the starting process of getting ready to divide. One may say that every cell is in interphase. Prophase is known when the nucleus begins forming chromosomes with the DNA present. By the end of this phase, one can clearly see separate chromosomes. Metaphase is visible when the chromosomes begin to align in the middle of the cell. This alignment is needed for anaphase to start. Anaphase is when the chromosomes split in half and the cell begins to divide into two. Once anaphase is finished, telophase begins. Telophase is basically just the finalization of the dividing of the cell into two separate operating cells. Once all these stages are finished, interphase begins again. I would very much love to go into greater detail of the cell with you all, but alas, I have not the time. With that being the end of my time, I am confident that I have given you enough information to have, at the very least, a basic understanding of the cell and how it works. By all means, if you have any questions, email us at info.with.a.voice@gmail.com. Would be great to hear from you all. And once again, this is JP Murray from Methods May Vary, a channel of InfoVoice. Looking forward to hearing from you all. Cheerio!